I want to address one question that one of your classmates, Nicole, asked me on last class if I could address this. This is from the chapter 6, the previous chapter. This is the first part of the question number 5. And basically, I can see what is the confusion, and I will explain a little bit. So the question was, for which of the following types of membrane transport is the rate of transport directly proportionally to the concentration of solid? So there is more than one answer. And before I go through the answers, I just want to remind you, right? There is basically 3 ways that biomolecules can pass the cell membrane. Recall that the cell membrane, it's a biphospholipid, have phospholipid by these 2. So the components inside the cell membrane, it's a nonpolar environment. Some of the biomolecules, they do have characteristic of nonpolar, right? We're talking about molecules like derivatives of cholesterol, some hormones, etc. So because of the chemical structure, they can easily pass without any help through membrane diffusion, right? So they don't care about nonpolar because they actually can interact freely. So this type of biomolecules can go directly through the cell membrane through membrane diffusion. Remember, through high concentration, upgrading concentration to low concentration. There are other molecules, as you can imagine, ions or biomolecules that are nonpolar. So they cannot pass freely. So they will need or require certain proteins. And in the previous chapter, right, we talk about different type of proteins, and some of the proteins are actually proteins that are in the cell membrane. So, and they actually help, those are channel or carriers, some are channels, some are carriers that can help some biomolecules to pass through. Just I emphasize a lot in the lecture the difference between the passive transport and the active transport. The passive transport, if you recall, it can transport ions like potassium, etc., without the need of external energy, like in active transport. And how this molecule pass through, again, is through diffusion, right? So from high concentration to low concentration. However, in active transport, you will need an external, as you can see here, source of energy, because most of the time is not through high concentration to low concentration, but against the concentration. And that's basically the way, right? It's high and low. In the case of active transport, it's against the concentration. The confusion, and I have to say that I did not emphasize what is the difference between channel and carrier, right? Just to show you, the passive transport, which is from high concentration to low concentration, there's 2 types. You can have a channel and you can have a carrier. The only difference is how these proteins, the conformation of the proteins. Here, you can see the channel is literally like making a hole in the whole cell membrane. In this cartoon represents, I think, very clearly. So it's like making a hole between the whole cell membrane. So the ions, we make the examples of the potassium channel ion that basically make a hole and it can go freely, right, through that hole, let's say. The carrier protein, it makes the change, the conformation, and it's like, imagine these revolving doors that one person go, right, and then pass through this revolving door until it goes to the other side. So imagine the carrier is like that. It makes a change in conformation, get the biomolecule or the ion that it's passing through, and then close it again. So it's not the completely whole of the channel. So I guess the confusion here was just the difference between the channel and the carrier. And actually on the PowerPoint, you have this illustration that is part of this figure that is 6.3a. And there's another part that I did not include and I did not emphasize, but the part B of that figure is actually that explain what is the difference between passive transporter that is a channel and the carrier. And here you can see that the translocation rate, so meaning when one molecule passes to another, how it's correlated to the concentration of the solute, which is basically what the question was asking. So in simple diffusion, in passive diffusion, I mentioned that these biomolecules, they can easily pass through because they are nonpolar. It's proportional, the concentration of the solute and the translocation. The same for the channel. However, in carriers, because of the conformation, there is a point where these type of proteins can be saturated with that specific biomolecule or ion, and they're actually similar to the enzymes, right? That there is a point that they get saturated with whatever, in the case of enzyme, is a substrate. In this case, will be an ion or biomolecule. So as you can see here, this is not proportional to the solute. So and I hope, Nicole, that was the confusion for you, because I saw that you select simple diffusion, which was correct, and you were asking why is passive. I know it's passive diffusion, but why is not carrier? And basically that's the answer. I did not emphasize that on the lecture, and it's just that difference. So what I'm gonna do is, if you guys, for that specific question, if you answer passive and one of the simple diffusion and one of the passive, either channel or carrier, I will give you the points of the homework. Okay.
Professor, yeah, that was what I was confused with. Thanks for going over that. But I just want to confirm. So you're saying with a carrier, at a certain point, the carrier protein will become saturated with the solute?
Yeah.
Okay.
You can see here, I don't know if you have access to the book, but that was the B part of this illustration that I put on the presentation, that it show you how is the translocation rate versus the solute. And you can see here the graph. This hyperbolic graph means that it gets saturated. Okay. Versus here. So actually it's faster when it's diffusion through a channel than diffusion through carrier. Okay.
Okay. Thanks for clearing that up.
Okay. Professor, this was from last week's homework, right?
Yes. It's just that Nicole asked me to go through, and I thought it was a good way for everyone. And I just want to emphasize that I did not make clear difference between channel and carrier, but now it's a good opportunity. But I make the emphasis, and I hope you have that clear, the difference between passive and active transport. Okay. Yeah, it's from last week. If you guys, I'm gonna go through the homework. If it was said to answer, if you have one or the other, I will give you the credit for that. Okay. So let's continue with the topic of enzyme. I mentioned that we're gonna finish with enzymes today. This is the third of 4 topics that will be included in your second exam, which is November 17th. And basically, we cover at the beginning the type of enzymes, how enzyme doesn't change the delta G, which is free energy. There's nothing to do with thermodynamics, right? Remember in chapter 2, we covered thermodynamics, but Enzyme, what it does is it deals with the transition state, right? It deals with the rate, which is the velocity, how fast a reaction can occur. Instead of taking years, they will take in seconds, right? And one of the characteristics of enzyme is that they don't change the product, they don't change the substrate, they don't change the product, they don't change the thermodynamic, the delta G, the itself, the enzyme that it shouldn't change. It will make a conformational change during the enzymatic reaction, but after that, the enzyme should be intact, right? In order to start another enzymatic reaction. Okay, and then we stop here, and this is basically the Michaelis-Menten plot. We can see here that the enzymes, there's going to be a point. We can see here the initial velocity of the rate versus the substrate of the concentration of the substrate. That there is a point that this curve will be this hyperbolic curve. It's showing you that the enzyme will reach their maximal velocity. So it will be saturated of the substrate. So it doesn't matter if you put more substrate, because if you don't increase the enzyme, your enzyme will be completely saturated. And you can see here in this illustration, the low substrate. This is the red represent the enzyme, the blue represent the substrate. And then this is at the beginning, right, in the initial velocity. And then it will reach when all the substrate find the enzyme and make the enzyme substrate complex, that there is a point that there is no more enzyme available. Therefore, the maximum velocity will reach the maximum velocity. And then we talk about what is a Km, which is basically the Michaelis-Menten constant. It's a concentration of the substrate when you have half of your Bmax. So if my Bmax is 1, half will be 5. So if I make a line here, then I will find the concentration of the substrate, and that will be the Km or Michaelis constant. We talk about how that represents the affinity of the enzyme to the substrate to the enzyme. Is an intrinsic property, so it won't change if you add more enzyme or substrate, because it's something that is intrinsic. We mentioned that the higher the Km, that means lower affinity. So it's similar to the concept when we discussed the Kd, which is Kd is dissociation constant for proteins. Here we're talking about enzymes, okay? And I mentioned that many drugs are inhibitors of enzymes. So we are looking, looking for a Km or Kd that is low, because that means that you require less amount of that substrate in order to block 50%, in this case, to get 50% of your velocity. Okay, so the lower the better, basically. And I mentioned, well, the Michaelis-Menten equation is here, is the initial velocity equals Bmax. This is the Bmax when all the substrate will find the enzyme, right? And then we have the concentration of substrate divided by the Km plus S. This is the Michaelis-Menten equation. I mentioned we are not going through the derivation of this, but if you are curious, I put a link, and you can find the derivation anywhere if you're curious about how exactly is that we get to this. Okay, and this is the representation, again, showing that you have two different concentrations of enzyme. The only way to increase the Vmax is that you increase the concentration of enzyme. However, as you can see here, the Km, the Michaelis-Menten, won't change because you increase enzyme. That is an affinity, that's an intrinsic property, and it's a property between the affinity of the substrate and the enzyme, okay? So here again, recall Km is the concentration of the substrate when you have a substrate when you have half of the Vmax. Are we clear here? We're good? Okay, perfect. Perfect. So then we have something that is called Lineweaver-Burke equation, and it's the same. It's just the double reciprocal plot of the Michaelis-Menten, and it's the same data. The only thing is it is more easier to get the Vmax and Km because the previous one, there is a point that the Vmax reaches the Vmax, but then you have to estimate it. With this other graph, you can get the data exactly, the exactly data. So it's easier to find the numbers, right, for Vmax or for Km in a Michaelis-Menten plot. Okay, so this is called Lineweaver-Burke equation, and as you can see here, is a Michaelis-Menten, but it's just the reciprocal plot. So Vmax here, it will be 1 over Vmax over initial velocity. We have in the X 1 over minus Km, which is a Michaelis-Menten constant. Well, it's a substrate, but then you can get the Km, so it's more precise. But it's the same data. And you can see here that actually the slope of this line, you can find the Km over Vmax, and it's some data that you can use. We will do some problems later. And here you can see one versus the other, right? It's the same. It's just one is it show you the reciprocal plot, right, from one another, but it's the same information. Okay. Questions here? So here we have in the X the concentration of the substrate, and in the Y the velocity. Okay. So velocity is in molar second, and in the substrate is just molar, but it's exactly the same. It's just the reciprocal. So another important details that you should know are how enzymes are compared one over versus the other, right? We know and we're clear about the Km, which is the affinity of the substrate with the enzyme, but that you cannot compare two enzymes, right? Just because of the Km. So there's other parameters. One of them is called Kcat, and this is actually also the Kcat is a turnover number. So how many molecules per second an enzyme can actually make. And just in your notes, please correct the following. In the notes, it says catalytic efficiency. Please cross efficiency and put catalytic rate, which is basically the Kcat. It is also known as a turnover number. Okay. And I ask you to remove the efficiency because there's another term. If you, the way to actually look for the efficiency, catalytic efficiency, is using Kcat over the Km, which is the Michaelis-Menten constant. And that's how catalytic efficiency is represented for enzymes. So you can compare two enzymes. Imagine you have the same substrate, different substrates, sorry. Although enzymes are very, very specific, right? They're very picky with the substrate. Sometimes they can have two of the substrate. The way that you can compare which one is more efficiency with the depends of the substrate is with catalytic efficiency. And we're gonna see an example. So just correct that. We have Kcat, which is turnover number, and we have catalytic efficiency, that is basically the ratio between Kcat over Km. Questions? Maybe when we go over the examples, you can see where you can use these two parameters, okay? So if you actually substitute the K-cat in the Michaelis-Menten, K-cat is actually Vmax over the enzyme, the total enzyme. You can have then, just by substituting that in the Michaelis-Menten, you can have this graph, just in case you have the K-cat number. K-cat multiplied by AT, because this is the Vmax. Recall that this is Vmax in Michaelis-Menten equation, times substrate divided by Km plus substrate. So it just, you substitute that in the Michaelis-Menten equation.
Professor, one question.
Yeah.
What is the ET again?
Sorry? Enzyme, total enzyme.
Okay.
Total amount of enzyme. So just make sure to correct this because the efficiency is actually K-cat over Km, okay? Clear? So? Okay. So the turnover, which is the K-cat again, it will give you the information about the maximum catalytic rate, but it won't give you exactly the efficiency. That's why you need K-cat over Km in order to know the efficiency for that specific enzyme. So when you can, if you want to compare which enzymes are more efficiency compared to another, you just look at the ratio between K-cat of Km, which is a specific constant, and the higher the number, the more efficient is that specific enzyme. Okay.
Can you go back to the previous slide?
Here?
Yeah, for ET, it's saying that total amount of enzyme in the reaction is E plus ES.
Mm-hmm.
But down here, it's saying K-cat is ET times S. So what is ES?
Say, can you repeat it? ET is the total amount of enzyme.
So there where it's saying total amount of enzyme in reaction, it's saying E plus ES. But
Is the enzymes and the substrate. It's actually just the total, let me remove this. It's total amount of enzyme. So that you don't get confused.
Yeah, so.
Total amount of enzyme. That's it.
All right, so down here where the subset for the equation.
Mm-hmm.
So it's ET times S. And then on the bottom half, it's Km plus S, right? It's K-cat times ET times S, and on the bottom it's Km plus S.
Mm-hmm.
Okay, just making sure.
Yeah.
All right, thank you.
So you just substitute in this, right? The Michaelis-Menten constant. The Bmax, right, is K-cat times ET, which is total enzyme, right? Here. So it's basically the same Bmax. So this is divided out, so you have the K-cat times ET. Okay. Who asked me? Excuse me? Can you repeat? Next time that you mentioned, if you can say your name so I know who is talking, because it's very hard for me. I cannot see anyone. I just hear voices.
Yeah, this was Ryan that was talking before.
Ryan, are you clear? Are we clear?
Yes.
Oh, perfect. Okay. Well, great. And make sure to correct this, okay? I don't want you to get confused because the efficiency is basic. When you mention efficiency is because you're using K-cat over Km. Okay? The S is the substrate. S is the substrate. And recall that this is concentration of the substrate. Okay? We're going to do some exercise. Maybe it's going to be clearer. So talking about the K-cat and the turn K-cat and the efficiency of the enzyme, let's do the following example. Using the table, answer the following. The information, we have different enzyme. We have the substrate. We have the kcat, which is the turnover, how molecules per second. You can see the units. We have Km, which is the affinity of the substrate for the enzyme. And well, it's molar because it's a concentration. And then we have the efficiency, which is kcat over Km. This is efficiency or a specific constant. Which enzyme has extremely high turnover rate, maximum catalytic rate? And how is the enzyme catalytic efficiency, the specific constant, compared to others enzyme? So let's do the first part. From this list, which one has the high turnover rate, so the higher kcat?
Catalase?
Which one?
Catalase?
Catalase. Okay. Who agree or disagree?
I agree with catalase.
Who disagree? Why is catalase?
I said catalase because it has the highest kcat.
Correct. And that's why they're asking, right? What is the high turnover rate? Catalase, perfect. But, and then from the list, how is the enzyme catalytic efficiency compared to others? So let's compare catalase with others efficiency. Recall that efficiency is a kcat over Km, so we have to look at this number. Compare, it has the higher kcat, but about regarding the efficiency, what can we say about it?
It's actually not that efficient compared to the other enzymes on the table. The triose phosphate isomerase seems to be the most efficient.
Yeah.
Okay. Who else say? Are you guys agree?
Carbonic anhydrase also has a pretty high, it's almost twice as strong.
Correct. So basically we are confirming what we are discussing, right? Like not because it has a high kcat, which is a turnover, how many molecules that specific enzyme will turn into a product, right? And that doesn't mean that the efficiency is better. So that's true. And then if we observe fumarase, these two, this enzyme actually has two substrates, right? The same enzyme, fumarase and malate. From here, what is the maximum catalytic rate for each substrate and which will be the best choice of a substrate and why from these two? What do you think? Answer.
Malate.
Malate will be a better Malate, wait, complete your answer.
Malate would be a better substrate.
Why? And why?
Because it has a higher catalytic activity and it has a greater efficiency.
It has a higher efficiency. Who else say the same, like agree or disagree?
I think it's fumarate because it has a higher kcat over Km.
Okay, who agree with?
I agree on fumarate as well.
Fumarate, okay.
Fumarate, I say the same.
Fumarate. Okay, and who say malate? Joel, what do you think is malate? What is higher, 1.6 10 to the 8 or 3.6 10 to the 7? So who can say what's higher, 1.610 to the 8 or 3.610 to the 7? Esther?
1.6.
1.6 times 10 to the 8th is higher.
Yeah. So the better option for this will be fumarate because the efficiency is higher, right? And as you see, the KM is pretty kind of similar, but who can tell us about the efficiency of an enzyme will be the KCAT over KM. Okay, are we clear?
So what is the answer to A for number 2?
Fumarate.
I thought that would be B. Which
Sorry?
would be the best? Isn't fumarate B, which would be the best choice for a substrate?
That will be the best choice because efficiency is higher.
And then what about for part A? It says, what is the maximum catalytic rate for each substrate? Is that just saying the KCAT?
Yes, correct.
Okay, thanks.
Correct. So just make sure to differentiate if they ask you for turnover and if they ask you for catalytic efficiency. Okay? That's why it's very important. Please correct this part. Okay?
Professor,
Yeah.
go back, yeah. So what was the reasoning behind the answer again? If you could just explain it for number 2.
The fumarate and malate? Who can explain why fumarate is better choice in terms of substrate?
Fumarate is better because it has a higher efficiency than.
Correct. As you can see here, who will tell you the efficiency of an enzyme is the same enzyme, but you have 2 substrate, is KCAT over KM, which is a specific constant, and the higher the number, the better. That means that specific enzyme will turn more products of, it's more efficiency by doing, in this case, producing the product that it's going to produce. So the higher, the better. And the KCAT is just going to be the turnover, and not necessarily will tell you if an enzyme needs more efficiency or not. Okay, so the higher the number, the better. So you have to get in consideration also the KM, which is the affinity of the substrate with the enzyme. Okay. Okay. I think. Okay, so we can look at this short video. It's in the textbook, and then we're gonna start some of the problems. Oh, actually, let's start one of the problems, and then we put the video. So let's do this. So if KCAT of an enzyme reaction is 10 to the 2 seconds, negative one, and the Km of the enzyme reaction is 510 to the 6. Here's a. Then the specificity constant of the enzyme reaction is?
There are a couple of extra multiplications.
Yeah. Yeah, just is 510 to the minus 6. So just remove, it's not x-axis, it's 10, 810 to the 510 to the minus 6. It's a typo. So the specificity constant will be
You guys can answer?
Yeah. Do you have the answer?
I mean, I think so.
What is your answer?
I got
What did they ask you?
Oh, basically, if the KCAT of an enzyme reaction is 8 times 10 to the second, oh, what's the substrate? It's to the negative 1, and the Km of the enzyme reaction is 5 times 10 to the negative 6. So the substrate right there, I see S to the negative 1. Does that make a difference?
Does that make difference? What?
You see S to the negative 1, the substrate? I thought that was the substrate.
That's the units for KCAT.
That's the unit, right?
Yeah, the unit. Yeah, correct.
Okay. Okay. Cool.
Yeah,
well,
the typo is just 8/10 to the second. You know.
Okay. So in that case, I got 1.6 times 10 to the 8th. I don't know if I'm right.
Who else?
Yes, me too.
Got
I
the
also
same
got
too.
that, Bakar.
Yeah, I got the same too.
Oh, good. It's not just me.
Okay, yeah. So basically, it's just kcat over km, okay, which is the specificity constant. Just make sure you know what they asking, and that's it. So it was pretty straightforward. Okay, good. Okay, so what about this? An enzyme catalyzes a reaction. A goes to B, right? The enzyme is present at a concentration of 2 nanomolar, and the Vmax is 1.2 micromolar seconds minus 1. The Km of a substrate 8 is 10 micromolar. Calculate the initial velocity reaction, which is the Michaelis-Menten, right? The equation. When the substrate is at 2 micromolar, at 10 micromolar, and at 30 micromolar. So what are you guys gonna have to do? First of all.
You can use Michaelis equation.
You have to change the units first.
What units?
Nanomolars and micromolars.
Who say the same? A ver. Go ahead and use the Michaelis-Menten. The other, just double check. So what do we need for the Michaelis-Menten equation?
You need Vmax, Km, and substrate concentration.
Okay, so we have Vmax here, right? And then we have the substrate, which will be different for each, right? A, B, C, well, it's gonna change. And then we need Km. Where is Km? The Km is here.
10 micromolar.
So do we have to change units here?
No, because we don't need the enzyme.
Yeah. You don't need that. At least for this particular, you don't.
Can you say why again? I missed that.
Okay, so can you tell me what is the Michaelis-Menten equation?
Yeah, it's Vmax times substrate over Km plus substrate.
So we have Vmax is 1.2 micromolar, right? S minus 1. And then we have the substrate, let's say for 8 is 2 micromolar. So we're good with the unit so far. And then we have Km, which is 10 micromolar, right? So at least for this particular example, we don't need any change, right? Because we don't need, in the Michaelis-Menten equation, we don't need the enzyme concentration, right?
Okay, so that's just extra information.
Yes. Perhaps for another example, you will need it, but not for this. So who has the answer for A, 2 micromolar? Ryan?
Is it? Oh, sorry.
It should be 1, right? It should be 1 micromolar minus to S to the minus 1.
1. Who else got 1?
I got 0.6 micromolar.
For the first one? For 2?
Yes.
Or for 10 micromolar? Check.
I got 0.2.
For A, right? And for B?
Same, I got 0.2.
0.6 for B.
Correct. Correct. Just check. Maybe you put 10. And for C?
.9.
Correct. It was us adding, and just make sure you know what is the Michaelis-Menten equation. Max times substrate over Km plus substrate. And you have all the information. So far, what you need to plot in this equation, all of them are in micromolar. Okay, so it's 0.2 micromolars per second, 0.6, and 0.9. We good?
Yeah.
Okay, good. So let's keep going. What about this? Let's do an inspection. So you're gonna estimate, right? Although graphical methods are available for accurate determination of Bmax and Km of an enzyme catalyzed reaction, in this you can see the quantities can be quickly estimated, right? So estimate by inspecting the values of B initial velocity at increasing substrate. So estimate the Bmax and the Km of the enzyme catalyzed reaction for which the following data were obtained. So you went to the lab and then you get this information, right? You have different concentration of substrate, you have different initial velocity. What would be the estimate the Bmax and the Km based on this information? And you don't have to do any calculation. You can just take an estimate. So what will be the Bmax? Any volunteer? Nicole?
I'm a little confused right now.
Okay. So who is not confused? Who can tell me what is the Vmax? Ryan?
I'm thinking it's something times 10 to the 8th or something like that.
So first, okay. So first, we're talking about Bmax. So remember, this is B, right? And recall how is the Michaelis-Menten plot that it will reach. If you plot this information, you will have a plot, right? A plot that is hyperbolic, and then you will reach a maximum value, right? So recall that in the X, in the Y, you have B, and in the X, you have the S, the concentration of the substrate, right? Yes or no?
Yeah.
So then it would be 140.
So you have here, right? And then shoot, and then you have the concentration, right, of the substrate. Who say 140?
I said 140 because at 140 you have the highest concentration of substrate.
That's it. It's the maximum velocity that this particular example will reach. 140. Yeah, that will be the maximum velocity. So this is telling me all the concentration of the substrate, and at 140 micromolar per minute, we're talking about velocity. That's my Vmax for this data. That is. And what would be the Km then? What is the definition of Km?
Half of the Vmax, so it would be 70.
Yeah. So what will be the Km?
1 times 10 to the minus 1.
Yeah, that's it. That's it. Km, recall its concentration, and will be the concentration of your substrate when you reach half of your Vmax. And that's it. So three concepts that if you're clear, it's pretty straightforward, right? Nicole, are you still confused or are you good?
No, I'm good now.
Okay, but that's...
Professor, what is it? Professor, what is the KM?
The Km will be, remember the definition is the concentration of a substrate when you reach half of the Vmax. Half of the Vmax in this example, so half of 140 will be 70. Therefore, the concentration in 70 corresponds to 110 to the minus 5. Okay? Okay. Are we clear? Yeah, right. Hold on. Every time I I cannot move my every time I try to write on the presentation, for some reason my computer just it's I cannot move anything. Oh my god. Give me a second. Okay. Oh god. Oh, finally. Okay. Okay. Let's continue. And now I don't know how to okay. And that's okay. This is a little bit, try to do the following. It says, the protease papain, and then here we're using a different graphic, right? The protease papain, which can be isolated from papaya fruit, is a monomer with one active site per protein molecule. The activity of purified papain enzyme was measured at various substrate concentration, which is basically what you do in the lab, right? You measure different concentration of the substrate, so you get the Michaelis-Menten plot. And the initial velocity, right, it is on the following graph. Considering that the enzyme concentration was held constant in this experiment at 1.2 10 minus 7 molar, okay? Answer the following. Calculate the Km, calculate the Vmax, and here they asking you for the turnover, which is the Kcat. So here you have this, but just to have the same answer, here let's put minus 8 and the intersection of this in the X, and then put 4 on the Y. Okay? So try to find the Km, try to find the Vmax, and the Kcat for this problem. So anyone finish? What is the Km and the Vmax and Kcat? Oh, let me put you this. Remember that in the line waiver bird plot, you have the Km. It's the same, the Km. You will find it right on the X axis, and in the Y, you will find the velocity, but it's the invert, so just be careful with that. So the Km and Vmax will be straightforward, right? So who can tell me what is the value of Km?
The value of Km 0.125.
Okay, and who else has the same? Who was that, Hannah?
Yes, sorry. Hi, Hannah.
Okay. And what about the Vmax?
I'm assuming it would be negative 0.25.
Okay.
Yeah.
Who has the same? The same. And what about the Kcat? And here we can find the Kcat equals Vmax over ET, which is now you can use the concentration of the total enzyme, which is basically here. Okay, so yes, the Km was 0.125 millimolar. Okay.
And then negative 2.08 times 10 to the 6 would be the Kcat.
The negative, what negative? Why negative?
Don't know.
It's 1 over V, which is in this case, the intersection here is because it's minus, right? We are in the minus and the formula is minus, but here the intersection it's 4. So 1 over 4 is 0.25 millimolar minus minutes, which is the units for velocity. And the Kcat, so you will need the Vmax, which is 0.25, but then you will need the concentration of a total enzyme. And now you can see that because you have the concentration of a millimolar, now you will apply that. Oh, be careful with the units, right? Because we have molar. Do you notice that? Hannah, do you notice?
No, I didn't. Okay, so then I would have to just fix that.
So now it applies what, I don't remember who mentioned that, but yeah, like, oh, we have to change. For that particular, no, because they were not asking for the Kcat, which is when you use the concentration, the enzyme, the total enzyme. But here we use it, and turns out that it makes our lives a little bit more complicated, and it's giving the data in molar instead of millimolar, which is the units that we're using. Okay. So just to make sure to change that. And anyone have the answer for that? Not yet.
Is it 2083?
2083 for molar?
No, I did it wrong.
All right, so just the 1.210 minus 7 molar, it will be minus millimolar. So 1.210 minus 4 is millimolar. And just because Kcat is per second, you have to divide it by 60 seconds, right? Which is a minute. And that's it. You will have the correct units. That was the most difficult problem that you will encounter here. That's it. Okay. Just make sure to change the units and make the correct units according to whatever they asking. Remember, KCAT, yes, it will give you information, but they won't give you the efficiency in case they ask you to compare between two enzymes or one enzyme with two substrates. Okay? Okay. So now,
One question. This is an example of what again? There was a name for it. I forgot.
the KCAT?
No. This question. It was something. It's an example of something. I forgot the name. Where No. Okay. Line.
Plot?
Okay, I got it.
Yeah, the Lineweaver plot is the same information as the Michaelis-Menten plot. However, because of the way it's arranged, it's more precise. So instead of estimated, so those values that we found, 0.127, 0.27, will be difficult to get with just the Michaelis-Menten concept. It's the same information, right? It's just the way it's arranged. So I'm gonna put the video, then the video they show nicely all of this that we have discussed so far. And that video is also on the homework, and just make sure to practice. This type is if there's a problem in the exam, you know my exam, so I will ask you whatever we discuss here. Okay? So let me go ahead and stop here. And then six and seven.
Enzyme kinetics. Measuring the Michaelis-Menten kinetic parameters of an enzyme is how scientists characterize and compare enzyme-catalyzed reactions. The overall enzyme reaction can be represented by the following formula, with the enzyme E and substrate S reversibly associating, reacting, and rapidly dissociating into the enzyme and product P. If we graph the concentration of enzyme-substrate complex, and product over time, we would see an initial burst of product formation that corresponds directly to the first step of the reaction, enzyme-substrate complex formation. This is called the pre-steady state period. After the pre-steady state period, the reaction reaches steady state, where all enzyme reaction steps are occurring at a steady rate. This is the steady state period, and during this period, the substrate concentration declines steadily as product is formed. By plotting the change in product concentration during the steady state period and finding the slope of the line, we can calculate the initial velocity of the reaction. Initial velocities are important for finding Michaelis-Menten parameters. When we determine the initial reaction velocities for different starting substrate concentrations, we see two trends. In these experiments, the enzyme concentration and the volume of the reaction are both held constant, whereas the substrate concentration is varied. First, the higher the initial substrate concentration is, the higher the initial velocity. Second, although initial velocities increase, the increase is not as great at higher substrate concentrations than at lower ones, because at high concentration, we are beginning to saturate the binding sites of the enzymes in solution. For this reason, when we plot initial velocity versus initial substrate concentration, we get a hyperbolic curve that is characteristic of first-order Michaelis-Menten kinetics. Using this plot, We can now determine other important Michaelis-Menten kinetic parameters. The velocity that the curve approaches asymptotically is the maximum possible velocity of the reaction, or Vmax. If we divide the Vmax by 2 to find half the maximum velocity, the corresponding initial substrate concentration is known as the Michaelis constant, or Km. Increasing or decreasing the enzyme concentration will change the maximum possible velocity, but will not change the Michaelis constant. It's a constant property of the enzyme reaction. Another way to look at the same data is to plot the reciprocal of the initial velocities and substrate concentration. This converts the data into straight lines and is known as the Lineweaver-Burk plot. Lineweaver-Burk plots make it easier to obtain the Michaelis-Menten parameters. The y-intercept corresponds to the reciprocal of Vmax, the x-intercept to the negative reciprocal of Km, and the slope is the ratio of Km to Vmax. Another way to compare enzymes is to determine the maximum catalytic activity under saturating levels of substrate. This value, called the turnover number, is defined as kcat. The kcat for a reaction describes the catalytic rate, but it does not reveal the catalytic efficiency. In the case of the enzyme fumarase, the Km for fumarate is 5 times lower than it is for malate. Yet the kcat values for these two substrates are nearly the same. This results in a specificity constant for fumarate that is about 4 times higher for the enzyme than when malate is the substrate. This higher efficiency means that fumarate is the preferred substrate of the enzyme.
Professor, this is Nicole. Will that video be on instructional materials?
I believe the video is on, in the homework. I believe so.
Oh, okay, thanks.
If you don't have access to in the book, it's in the book. But if you don't have access, it's on the homework. So you can review that. So that very nicely summarize everything that we have discussed today. And now, as you can imagine, right, enzymes are proteins. And proteins are very sensitive to the environment and to the pH, obviously. It would change the environment of the pH and the temperature too, right? The pH, we know that it can change the ionization of certain amino acids. And if they are in the catalytic side of the enzyme, will be affected if you change the environment and also the temperature, right? You know that some are resistant to temperatures, but others are not, since our body will be 37, but perhaps other enzymes that are very resistant, they will need a specific temperature of 80. But if a protein that is an enzyme, the ideal temperature is 37, and you put it at 80, obviously it will be denatured and won't work anymore as an enzyme. And the pH too, so depends on the type of enzyme and the environment, whatever they do, the biological activity, for example, pepsin. Pepsin is an enzyme that is in our stomach. So the stomach, we have a pH, a low pH, 1.6. So therefore, for pepsin, the ideal pH for the enzyme activity will be very acidic, 1.6. But for others, right, any enzyme that work in other compartments of the cells where the pH, need a pH about 7, that will be the ideal, okay? So depends on whatever the enzyme, the role, then they will need a specific pH or temperature to have the activity, okay? So now we're gonna go another important topic of enzyme, which is the inhibitors. Enzyme, as you can imagine, are very well regulated. Either if the body doesn't need more enzyme, perhaps it will shut down at the DNA level, right? It won't create more enzymes, but also there are other types of regulation of the enzyme, right? And the regulation can be, they can stimulate the enzyme or it can inhibit the enzyme. As I mentioned, bioavailability is that how much enzyme it's in my body, right? If I don't need specific enzyme, it's just, you're not going to produce more dose of enzyme. Or perhaps another way to regulate the activity of the enzyme is through catalytic efficiency, through certain modifications. Here, if you don't need what, you just stop the process of creating the protein synthesis of that specific enzyme. Or if the enzyme is there, maybe you can do certain modifications. Some of them we have discussed this already. For example, dephosphorylation is a very important form of controlling the enzyme, right? When you phosphorylate it or dephosphorylate it. There are other mechanisms, but basically that's one of the examples. And the inhibitors, right? Inhibitors, that they can inhibit the enzyme, either reversible or irreversible. There's the two types here. We have irreversible, as the name implies, once this inhibitor binds to the substrate, to the enzyme, what's going to happen is will kill the enzyme. It will bind so tightly. So they're going to form covalent bonds or very strong non-covalent bonds, and that the enzyme, that's it, that enzyme is not functional anymore. And one of the examples is, for example, it's aspirin. Aspirin, the way it works is it binds to this enzyme that is called cyclooxygenase. And those are responsible for creating or producing prostaglandin, which are associated with inflammation. So when you're feeling, when you have a pain, right, you get some ibuprofen, aspirin, and that's basically it's an inhibitor of this specific enzyme. That's why it reduces the production of prostaglandin, and therefore that inflammation, that pain is reduced. So that's an example of irreversible type of inhibition. That's why I mentioned that many of the drugs that are available are actually controlling the enzymes. The other type of inhibitors can be reversible, meaning once you remove the inhibitor, the enzymes are fine and they can keep going and going, right? So we have three classes. We have competitive, uncompetitive, and mixed. And each one will have specific characteristics. So we have the inhibitor, competitive inhibitor, as the name implies, it will compete for the binding site of the enzyme. So you have the substrate here. This is an enzyme in yellow, and the substrate will bind to the binding site, and you have a product. When you have this type of competitive inhibitor, it will compete for the binding site. So it will. It's similar. Obviously, the structure is so similar that it can actually bind to the binding site of the enzyme. Because for this type of competitive inhibitor, it competes with the binding site, but as long as you put more substrate, the enzyme will be fine. It's irreversible, right? So as long as you put more excess of the substrate, your enzyme will be fine. So it will prefer the substrate over the inhibitor. Some of the characteristics of the competitive inhibitors, there is no change of Vmax because once, assuming you put more substrate, you're going to be fine. So you got the Vmax, the maximum velocity as normal, but there is an increase of the Km, which is the association of the substrate and the enzyme. If it increases, that means it has less affinity, okay? Because this competitive is competing in the binding site. The other types of type of inhibitors, they won't bind in the binding site, but they bind to they will bind in another places in the enzyme. So again, this is the illustration. You have the enzyme, and the substrate will bind to the binding site, right? The catalytic unit. This type of competitive, either competitive or mixed, the inhibitor will bind in another place of the enzyme. It's allosterically will bind. Remember, allosterically means other than the binding site. Okay, so it will bind in a different place, not in the binding site. Because it's binding to another place, obviously there's going to be an effect of the enzyme. In the case of uncompetitive inhibitor, what's happening is it exclusively binds just if the enzyme substrate is already formed. So this type of inhibitor likes to bind once the enzyme and substrate are already bound. And because it doesn't bind to the same site, it can bind somewhere else and it will affect. What happened here is that there is a decrease in Bmax because it's affecting the enzyme, so it won't reach the Bmax, and it will decrease the Km. It will decrease because once it's bound, remember this will bind just because one enzyme and substrate are already bound. So it makes more affinity, the substrate to the enzyme. So that's why it decreases the Km. And the mixed inhibitor, there's several, but for this class, we're just gonna focus on the non-competitive inhibitor. The non-competitive inhibitor, as the illustration show, it can either form binds to once there's no substrate. Recall that in the case of mixed and non-competitive, they will bind in another place, not in the binding site. So it's not gonna bind here where your substrate binds to, just the competitive. So for this mix, it can bind it's itself, the enzyme, or when enzyme substrate are together, okay? In the case of not competitive, there are some characteristics. One is that—
Professor?
Yes.
Can you repeat that for me for the mix? It binds?
The mix, as you can see in the illustration, it can bind either when the enzyme is itself or when it's already bound to the substrate.
Oh, okay. Thank you.
So what happened here is that it decreased the Vmax, so it will affect the enzyme, the velocity, the maximum velocity, but there is no change in the case of the Km. Okay. So those things, you should know what is the difference and what is happening, and we're gonna look at the graphic, and you should distinguish which one is competitive, uncompetitive, or mixed. Okay. Again, I mentioned this before. If it's competitive, it competes, and the name implies because it competes for the binding site. For the binding site is where the catalytic activity occurs. When the substrate binds to, that's the binding site. So this is just an illustration, right? The substrate is in the binding site, but this specific inhibitor will bind to the binding site. Again, there's no change in Bmax, but there is an increase in Km. When we look at this, the graphic, you can see here, there is no change in Bmax, okay? There is no change. You can see the intersect is the same when you change the concentration of inhibitors. However, the Km will change. Okay, here you can see better. For you, it's the same information. Is the same information. Is there any question? Or is me? Can you please mute? I don't know who is on mute, unless you have a question, but I cannot. Thank you. So you can see here how is the graphic, and you should know this, how is a graphic for competitive So here is no inhibitors. Again, there is no change in Vmax. You cannot appreciate here. That's why we need the other type of graph, because here is clearly that the Vmax is not changed, but the Km is changed. Okay. An example, a real example of type of competitive is, for example, methanol and ethanol. Methanol is toxic, so for any reason, someone, I will say by mistake, right, we get a methanol, which is toxic. One of the treatments in the hospital is actually at the competitive substrate, which is ethanol. Both are competing for the binding site. So you add excess of ethanol in a controlled way, obviously, in order to remove, right, to compete with the methanol, which is one of the competitive substrate for this particular enzyme. So both are very similar. They bind and compete for the binding site. Another type of example is this type of some drugs, in this case is HIV proteases inhibitors. Obviously, because it competes for the binding site, the chemistry between both, it should be very similar, so they can bind and bind perfectly to the enzyme, right? Just some examples. So in competitive, I mentioned one of the characteristics is that the enzyme, the substrate, sorry, the inhibitor will bind just when the enzyme substrate is already formed, as you can see here. And this is the same, it's just different illustration, but it's the same concept, right? Once the enzyme finds the substrate, then this type of inhibitor will go and bind in another place allosterically. So the type of line is here. You can clearly see that the Vmax change and the Km change. Okay. So this is uncompetitive. It will change both Vmax and Km. And the mix, you can either go and bind to the enzyme when it's itself or when it's mixed, where it find the substrate. It can be any of the two. There's several, I'm gonna go here. There's several types, but we're gonna focus on the one that is categorized like non-competitive inhibitors. And you can see clearly that there is no change in Km, but there is a change in Vmax, okay? It changed. Okay, so just focus on this. We're not covering this, we're just covering the non-competitive inhibitor. Okay, so we're covering competitive inhibitor. Make sure what are the characteristics and how is the graphic. You can see here there's no change in Vmax, there's a change in Km. Uncompetitive, it changed both, and non-competitive, these three type of inhibitors. Okay. So if I ask you what type, right, of in the line waiver plot here, which is what type of inhibitor are we talking about? Let's see if you are still awake. Just by looking up of the plot. Okay. I'm gonna wait 20 more seconds. So it's a non-competitive type of inhibitor, and we have a mix here of the results. We have a mix, literally. 37 say competitive, 29 uncompetitive, 30%, 34 non-competitive. So just make sure to go and review the lines and what's happening and what is competitive and non-competitive. Okay. So that was non-competitive. There is a
Professor, why is it non-competitive?
because according to what we discussed before, there is no change on Km and there is a change in the Vmax. So if you go back to each one, see, this is the characteristic of the non-competitive. Okay, the competitive, again, it won't change the Vmax, but it will change the Km, right? As long as you put more extra substrate, you will be fine. I mean, the enzyme will be fine. The uncompetitive, it will change both, and you can see here. And here, I mentioned that we're just going to focus on the non-competitive, and this is the characteristic for the non-competitive. Okay. Okay. So that's about inhibitors. I know it's a lot of information. You have to go back and take a look. What are the illustrations? To me are very, very clear and very good. Like you can see, well, competitive, it will compete for the binding site. It won't change Vmax because the enzyme will be fine as long as you put more substrate, but it will change the Km, right? Non-competitive, etc. So check what are the characteristics. They're very clear characteristic of each ones. Okay, so the regulation, we mentioned this before, right? That are very regulated because it depends of the body, that if you need it or not. It can be non-covalent modification or covalent modification, allostatic or non-allostatic. The modification can be allostatic. Again, recall allostatic means allos mean other, so meaning it will bind in another place of the enzyme, but it will change the enzyme to be either positive or negative, right? It will reduce or increase the enzymatic catalysis. And here, some of the enzymes are not specifically with the hyperbolic graph, which is the Michaelis-Menten, but they are like the S, a little bit more S. So these will represent cooperativity. If you recall, we mentioned cooperativity on hemoglobin. Hemoglobin is not an enzyme, but it's a good model for a protein that presents cooperativity, which means that when one of the substrate binds to, in the case of hemoglobin, is oxygen, it will help to, or it will modify the other subunits of that specific protein. The same for enzyme. So for example, here, you have the R state, which is basically the same. The R, recall in hemoglobin, it was the relaxed state. You have affinity, and you can see here that the K, the concentration to reach the Vmax is lower than in this other graphic, which you need a lot of substrate in order to reach the Vmax, right? So then this side, the red or pink, is the T state because it's the tense. So this resembles actually the oxygen saturation curve. So some of the allostatic molecules that will bind to the enzyme will either make the enzyme more high activity or less activity, depends. That's a way of regulation. We have feedback inhibition. Again, allostatic regulation means that it will bind in a different place, not in the binding site, in the catalytic site, but in another place that will make the enzyme to be activated or deactivated. So the feedback inhibition means that in signaling, let's say this is a signaling, so you will make A, B, C, and the final product will be D. Turns out that D, the final product, will be the inhibitor of, it will give a negative feedback for the first enzyme. So obviously, if you want to create D and there's a lot of D in your body or in the system, why do we have to go do more D? So D will block enzyme 1. This is a type of regulation, and this is a real example. This type of enzyme ATCase make the pyrimidines, which is the nitrogen base found in nucleic acid. If there is a lot of CTP, right, in the body, in the system, why do I have to do more? So therefore, CTP will go and block this specific enzyme, okay? By the opposite, if you have a lot of ATP and you have to produce more CTP, therefore ATP is a positive feedback, okay? So that's type of allostatic regulation in enzymes. This is the same example. The final product is cytidine triphosphate, CTP. This will be a negative regulator because why do I want more if I already have plenty of that? So it's a great system, right, to regulate the enzyme. So it's going to go and say, hey, let's stop this reaction because we don't need more, okay? And this is just showing you have the control. If you have ATP, CTP, which is the final product, you have the red, which is inhibiting, right? Showing that you have a less activity of this particular enzyme. If you have more ATP in this particular example, that means that you have a positive regulator, okay? And one of the final things is covalent modification. We have mentioned this over and over, right? That some of the modifications are phosphate with kinases. Some of the amino acids are serine, threonine, and tyrosine in the OH, in the hydroxyl group. Other groups also are methyl, acyl, adenyl, etc. I don't know if you have heard DNA methylation, but there is a way to control the gene expression through methylation. So there are groups that you will add or remove in order to control, in this case, the enzymes. And the phosphatase, well, is the opposite of the kinase. It will remove the phosphate. And so, for example, insulin, which is a huge cascade. Next week, we're going to talk about cell signaling. It's a huge, is the way how cells communicate. So there's a lot of signaling pathways. And all of that, obviously, through enzymes. And 2 modifications, you can turn on or off a whole cascade, right? If you're eating a lot and you have a lot of sugar, that means you have to activate certain pathways that if you have the opposite. Here is just showing the phosphorylation in the last one. In this one, for example, when we go in the morning, right? Assuming you didn't wake up in the middle of the night, you starve for 10 hours or so. When you wake up, the levels of glucose, the glucagon, the hormone, that means that your body will need glucose, right? You need a source of glucose in your blood. Therefore, this hormone will activate and give the signal in order for this enzyme, this is called phosphorylase kinase, to be phosphorylated. And that's phosphorylator. It will activate the pathways and the metabolism that is necessary to get the glucose that your body needs. So it's very, very cool, but depends on what you need. That simple phosphorylation can activate many of the pathways. And that's a way for regulating enzymes. And that's it. It will be our last slide for this topic. And I have a video if we have time. But basically, some enzymes are actually inactive. So they are inactive precursor protein. The example is a pepsin. Pepsin, when it's in the body, right? Pepsin, recall, is one of the examples that it's a protein that it's found in the stomach. So the pH for pepsin is very acidic, 1.8. So in the body, when it doesn't need to be active, is in a form of pepsinogen. Inactive is called zymogen. Zymogens are enzymes that are not active. So in the case of pepsin, it's inactive in our body. Once it goes in the stomach, where it actually do the biological activity, then there will be activation. It will actually do a proteolysis. It will cut the end portion of that specific enzyme. So then now it's going to, instead of being pepsinogen, which is active, it's going to be pepsin, which is active. Okay, so zymogen is just the terminology. When you heard that, that means that it's a protein that is inactive. So something needs to occur. In this case, it doesn't need the end terminal of that portion, and then it's going to be active in our stomach. Okay.
I have a question. Are zymogens usually proteins that are dangerous to the body? Because I was reading about them, and the reason why they get activated is because they can only work in certain places, like for instance, stomach acid, like because otherwise it will like destroy like the body.
I don't know if it's dangerous. Obviously, they won't be functional because, for example, pepsin, it needs the pH 1.6 in order to be functional, like to do proteolysis. I cannot answer that. It will be dangerous. Where do you read that?
So like I read that like zymogens, the reason why they get activated at certain pHs is because if they're active all the time, like stomach acid, it can like eat away at tissues if it's not where it's supposed to be.
Okay, let me find out about that. I know if they are not active, they're not active, right? They're not, they don't doing their job. For this, it needs to be active. Therefore, at pH very low. So I don't know. I cannot tell you that those are dangerous for the body. But I know they're not active. So let me find out and then we discuss that.
Okay, thank
That's
you.
kind of an interesting topic, though. I will read about it too. I'm just gonna look it up and see.
So they were dangerous to the body?
Yeah. You know, with everything has an advantage and a disadvantage in the body. So I'm pretty sure there's some type of disadvantage that it does to the body.
Yeah, I don't think I don't think dangerous might be the word. I think it's just like
after effect of stuff.
It's like it has to be like you can't just have like pepsin all over the body, right? Like you can't because otherwise it will like kill like or eat away certain cells that it's not supposed to be eating at. That's why it only gets activated under certain pHs. Because like for instance, it only works at pH 2, so it's activated in the stomach where it won't eat away. That's what I meant. It's not like dangerous. Like oh no, it's gonna like
No, I would say our body is just I don't know magic. No magic. There's no magic, but perfect. I mean, to me, that regulation. All of this is just so amazing. I mean, I wouldn't say the word dangerous because no, but it's so smart, right? Because it will be activated when it needs to be activated. It needs to go in the stomach and degrade it as a protease, so cut peptide bonds of the food that we get, right? So yeah, interesting.
Yeah, thank you, Professor. Sorry for like the confusion. I was just like wondering.
You got scared, like, wow, it's dangerous, but good. Okay. So questions, let me see the time. So that will be our last slide for, oh, well, we have time because the video is 2 minutes or 3 minutes. And also it's on the homework, but it mentioned how is the regulation of enzyme, which is amazing to me, how the body is so smart, right? When you need it, you need this, that. So let me share this and any questions or comment? Seven.
Many enzymes need to be tightly regulated to ensure that they are active only when and where they are needed in our cells. In this video, we will explore 2 examples of how specific enzymes are regulated. The bacterial enzyme aspartate transcarbamylase, or ATCase, is an example of an enzyme that is regulated through an allosteric mechanism. ATCase is responsible for an early step in the production of the nucleotide cytidine triphosphate, or CTP. Specifically, ATCase catalyzes the formation of N-carbamyl L-phosphate from carbamyl phosphate and aspartate. ATCase activity is upregulated by binding to ATP and downregulated by binding to CTP, the end product of the pathway. This regulation ensures that if enough CTP is already present, more will not be made. Note that ATP and CTP bind to a site that is distant from the active site of the enzyme and are thus considered allosteric regulators. When there is a high concentration of ATP relative to CTP in the cell, the regulatory subunits of ATCase bind to ATP. ATP binding stabilizes the active conformation of ATCase, called the R state. The conformational changes propagate from the regulatory to the catalytic subunits. In the active or R state, the geometry of the active site stimulates catalysis by bringing the carbamyl phosphate and aspartate substrates into close proximity with one another. Once the concentration of CTP has increased in the cell, ATCase will shift towards binding CTP rather than ATP. CTP binding results in shifting the conformation of ATCase toward the inactive T state, effectively shutting down the enzyme. This is an example of feedback inhibition. Protein cleavage, or proteolysis, is another way that some enzymes, particularly proteases involved in digestion and blood clotting, regulate their activity. Pepsin is a digestive enzyme synthesized in the stomach that aids in breaking down food by catalyzing the hydrolysis of peptide bonds. This results in converting long protein chains into smaller peptides. Pepsin is first produced as an inactive precursor, or zymogen, called pepsinogen. In pepsinogen, the active site of the enzyme is blocked by a 44 amino acid N-terminal protein fragment. The acidic environment of the stomach causes a conformational change in the pepsinogen that results in it cleaving its own N-terminal segment. The active site is now accessible. These examples illustrate only 2 ways that enzymes are regulated to ensure that they are active at the proper time and place within cells.
Okay. So that's how some of the forms that the enzymes are controlled, and zymogen, well, they are inactive. So whenever the body needs it, then will be active. Okay. Questions? If there is no more question, well, I will see you guys on Monday to cover, that will be our last topic, cell signaling, before your second exam. Okay?
Professor, thank you and have a good night.
You too, have a good night.